Welcome back friends, welcome back to another short video tutorials on developmental biology, Gilbert. Today we'll try to understand an interesting topic on developmental biology and the development of human brain and cognitive development during the course of evolution. There's a one gene that is given here, SRGAP2 gene, whose duplication is associated with the increment of the acumen of human IQ and cognitive development. So the cognitive development is also associated with the DSCAM protein that we have already discussed in previous video, that this DSCAM protein present in the dendritic region of a neuron doesn't allow the communication between the intradendrites of a same neuron. Now, duplication and divergence of human SRGAP2 gene. The SRGAP2 gene is found as a single copy in the genome of all mammals. If you study any other mammals that is not human, like dogs, cow, etc., they have only one copy of the gene except human. So in the lineage giving rise to human, duplication events give rise to 4 similar versions of the gene. So gene duplication is associated with this increment of neuronal flexibility, designated as A2D. The ancestral gene, let me scroll down, SRGAP2A with a minor contribution from SRGAP2B and T enable the maturation of dendritic spines. That is very important for our intellect. Protuberances on the surface of the neurons. SGRAP2C is a partial duplication, and the variations and product inhibit SRGAP2As. It is a slowing dendritic spine maturation and promoting neural migration. This partial duplication may have allowed for the evolution of longer maturation time and greater flexibility in the human brain. Thus, these genes are homologous between species as opposed to member of a family being homologous within a species. So, genes that are homologous between species are called orthologous. This is a very important information. One of the most important gene duplication event in the human evolutionary lineage of evolution may have been duplication of SRGAP or SRGAP2 gene, a gene that may have enabled the expansion of the human cerebral cortex. This is what the evolution supports. The protein encoded by this gene is expressed in mammalian brain cortex and appear to slow down cell division and decrease the length and the density of dendritic process. However, humans differ from all other animals, including chimpanzees, by having duplicated version of this gene twice. Moreover, the second duplication event was not complete, so one of the newly formed gene is only a partial duplicate. Gene duplication is also seen in the pseudogenes, but those genes are not functional. This partial gene duplication in product a truncated version of SRGAP protein, SRGAP2C, that is also made in cerebral cortex and which inhibit the activity of normal SRGAP2 made from the complete genes. As a result, cell division in the cerebral cortex continued for longer period of time and the dendrites are larger with more connection. This is how there is a synaptic flexibility and development of human intellect. Based on genomic evidence, the genes duplication event are calculated to have taken place about 2.4 million years ago. This is what the time frame data. This would be about the time of Australopithecus. The increase in primate brain size. The primate brain size increased due to this gene duplication and the first known use of the tools Tyler Smith and Liu 2012. So this is the short video on gene duplication and how it is correlated with the development of human intellect and brain cerebral cortex. In the coming video section, we'll discuss about deep homology. We'll read the book and understand the statement. Thank you for watching.